We're going to take a closer look at the process of exchange in capillaries. We're going to use the continuous capillary as our example because this is the most common type of capillary. And the reason why we're doing exchange, don't forget, is because the job of a capillary is to provide things like oxygen, nutrients, and water to the cells and tissues of the body. Likewise, the cells and tissues of the body produce waste like carbon dioxide and urea and sometimes excess water that needs to be removed from the cells and tissues and put into the bloodstream. So that's why we're going to be doing capillary exchange. Now let's take a look at the how of capillary exchange. We really have two ways that we can do capillary exchange: the transcellular route and the paracellular route. The transcellular route is shown with the red and blue arrows, and the paracellular route is shown with the yellow and green arrows. And you can see they're a little bit different. In one case, we're moving across the cell, and in the other case, we're moving between cells. Transcellular means to go across the cell. Paracellular means to go between the cells. With the transcellular route, we can really only do this via diffusion. And diffusion is when substances move from an area of high concentration to an area of low concentration. And we can't do that with a lot of stuff. So there's going to be very small amounts of diffusion occurring across capillary walls. A lot of the exchange is going to occur through that paracellular route. And because there's a lot occurring here, we call this bulk flow. With the paracellular route, what happens is water and small things dissolved in the water are going to move through those intercellular clefts between adjacent epithelial cells, making up the capillary wall. This is dependent on a pressure gradient, and we're going to talk more about these two things in more detail. Take a quick second to quiz yourself to see if you understand paracellular and transcellular exchange. So we have 4 options on the right-hand side of the screen. 2 go with paracellular and 2 go with transcellular. See if you can figure out which is which. Pause the video. When you're ready to check your answer, hit play. So here's your answer. Paracellular exchange matches with things moving between the cells of the capillary wall and being driven by a pressure gradient, whereas transcellular is referring to things moving across the cell membrane of the capillary cells, and it's driven by a concentration gradient. Diffusion is the driving force behind the transcellular route. Let's take a closer look at diffusion across cell membranes. There are 2 main types of diffusion that can occur across the cell membrane: simple diffusion and facilitated diffusion. Simple diffusion occurs when the substance is moving right across the phospholipid bilayer of the cell membrane without any type of protein in the cell membrane helping transport the substance. This is not an easy thing to do. Cell membranes, specifically the phospholipid bilayer of a cell membrane, is meant to be a barrier to almost everything in the body. So only a few things can actually cross this phospholipid barrier. That includes water, gases, and anything that's hydrophobic. The other type of diffusion is facilitated diffusion, and as the name suggests, we will use a facilitator here. The facilitator is in the form of a protein. So there's a protein in the cell membrane that's being used to transport the substance. In both simple diffusion and facilitated diffusion, we're still using concentration gradient to cause the substance to move. So the substance is always moving from the area of high concentration towards the area of low concentration. It's just whether or not we're going right across the phospholipid bilayer or through a protein. So don't forget, with diffusion, we're going right across the cell membrane. We're using that transcellular route, right? But this does not account for the movement of large amounts of substances across the capillary wall. So for that, we need to take a closer look at the paracellular route and bulk flow. With bulk flow, we are going to be moving large amounts of fluid and dissolved substances across the capillary wall by going through the intercellular clefts. Keep in mind that bulk flow can occur going into the capillary and going out of the capillary too. Unlike diffusion, bulk flow is driven by a pressure gradient instead of a concentration gradient. So it's very dependent on the current pressures in and around the capillary. Some pressures push fluids into the capillary, and some pressures push fluids out of the capillary. And just whichever one is pressing hardest is the one that will win. There are two main pressures that will determine bulk flow. Blood pressure, also known as blood hydrostatic pressure, and then blood osmotic pressure. So blood pressure or blood hydrostatic pressure is just the pressure that blood exerts pushing on the walls of the artery or capillary in this case. It's similar to the pressure that water would exert on the walls of a hose. This is an out pressure, so it's trying to push fluids and dissolved substances out of the capillary. The other pressure is blood osmotic pressure, and as the name suggests, it will involve the process of osmosis. So don't forget, osmosis is just the diffusion of water towards the area of higher solute concentration. So water always moves towards the high solute. The most important solute here is the blood plasma protein albumin. So because the blood plasma protein albumin is found in the blood, it serves to draw water via osmosis into the capillary. So this is an inward pressure on the capillary. There are other pressures that can be involved with capillary exchange, but for simplicity's purposes, we're just going to talk about these two. When we look at a capillary, we see that the only pressure that really changes a lot is blood pressure. So blood pressure very consistently changes across the capillary. And as you can imagine, the blood pressure coming into the capillary is much higher than the blood pressure going out of the capillary. So the blood pressure on the arterial side of a capillary is around 35 millimeters of mercury, whereas on the venule side, it's at only about 18 millimeters of mercury. So that's a pretty good drop in pressure across our capillary. Blood osmotic pressure is at about 25 millimeters of mercury, and it does not change across the capillary because it's due to that protein albumin in the blood plasma, and that protein concentration is not changing across the wall of the capillary, so that pressure does not change. So now that we have some numbers to associate with this process of bulk flow, we can compare the numbers and just see which one's bigger, and then we know who the winner is. So when we look on the arterial side of the capillary, we see that the blood pressure at 35 is much higher than the blood osmotic pressure at 25. So because the blood pressure is an out pressure, that's our winner. So this means that bulk flow occurs going out of the capillary on the arterial side. The opposite will occur on the venule side of the capillary. When we look at the venule side of the capillary, the blood pressure has dropped considerably to about 18 millimeters of mercury. The blood osmotic pressure is still at 25 though. So that 25 is drawing water and dissolved solutes into the capillary. So this drawing in is higher than the pushing out. So on the venule side of the capillary, fluids and dissolved substances are going to enter the bloodstream. We can use the words filtration and reabsorption to talk about this process. What happens on the arterial side of the capillary is called filtration. So when that high blood pressure pushes the fluid out of the capillary, that's filtration. On the venule side of the capillary, when that higher blood osmotic pressure draws fluid into the capillary, we call that reabsorption. It's called reabsorption because you already absorbed these substances once in your intestines, so now you're reabsorbing them again. The substances that are moving via bulk flow are water and any small hydrophilic substances dissolved in the water. So this includes things like amino acids, glucose, ions like sodium, potassium, and calcium. So these things dissolve in the water, and as the water is moving in and out of the capillary, they will move with it. Larger substances like proteins are too big to fit through the intercellular clefts, and they cannot move via bulk flow. We generally don't move a lot of proteins in and out of capillaries anyway, and typically we will do so at capillaries such as our sinusoidal capillaries or fenestrated capillaries. So how do we regulate capillary exchange? Keep in mind that highly active tissues, like skeletal muscle when you're exercising, will require a lot more blood than tissue that's less active. Like when you're exercising, you're not using your stomach and intestines. So we need to be able to control the permeability of these vessels. And we're mostly going to do that by controlling dilation and constriction of arterioles and precapillary sphincters. Most of this happens through autoregulation, which means that we don't need the nervous system to control this process, and we don't need hormones to control this process either. So it's kind of cool. What happens is when a tissue is highly active, it's going to start to warm up. It's going to start to produce more carbon dioxide than normal. The presence of the carbon dioxide will cause the pH to go down, becoming more acidic. And there will be low oxygen in the blood of this highly active tissue because the tissue is pulling lots of oxygen out to make ATP. So it turns out that the arterioles and the precapillary sphincters are sensitive to all of these things. So arterioles and precapillary sphincters will dilate in response to higher temperatures, lower levels of oxygen, higher levels of carbon dioxide, and lower levels of pH. And then the reverse occurs when the tissue is no longer being active. So when the tissue is less active, there will be more oxygen present, there will be a lower temperature, there will be less carbon dioxide being produced, and the pH will rise and go back to normal. And again, the arterioles and precapillary sphincters are sensitive to this, and when they detect this, they will constrict and cut off blood flow through that capillary because that capillary is feeding tissue that no longer needs those substances. So that's the process of autoregulation.